Welcome everyone to the HUPO ECR webinar about the mystery of grant writing. We have three great speakers with us today. We have MacCoss, Jesper Olsen, and Rebekah Gundry. And our first introduction is going to be for Professor MacCoss, who originally got his PhD in analytical chemistry for focusing on the development of stable isotope and mass spectrometry methods. And then following his PhD, he did some postdoctoral research at the Scripps Research Institute under John Yates. And since 2004, Mike has been at the University of Washington with his own group, becoming professor in 2014. And he's continued to be a big name in the field, making profound contributions to proteomics research. So Mike, if you want to share your screen and introduce yourself.
Great. So I just want to kind of introduce myself very briefly. And one thing I think it's kind of important to understand in the way that I approach at least grants is that everything we predominantly focus on uses mass spectrometry. We're a mass spectrometry lab. That's what my interest in. That's sort of what the main things that kind of excites me. One of the challenges, of course, is actually trying to get funding to do technology development is always a tough thing. And one of the things that's really important, of course, is to then begin to think about what the applications of the products are. One of the reasons why I'm in a Department of Genome Sciences in a medical school is because of the applications of proteomics. One of the things I feel as an analytical chemist, it's outside of my expertise often on the biology and the biomedical questions of that, and working with really great collaborators in the context of the correct applications that you also can learn a lot about the biology from those collaborators as well is very important. I will also say that my background is very much in analytical chemistry. I think a lot about quantitative figures of merit a lot. That's basically a huge chunk of what my training was in. But I think in the context of grant writing, it's while it's very valuable to think about those things, and it's very important to know those things, and I think essential in the application of those things. I find that what is key to kind of selling at least grants in biomedically funded research is on the importance to the biological applications. So what I often think about is in the context of thinking about what the sample is. And so mostly these green boxes and these red boxes, and ways in which we can kind of try to improve the throughput, improve the spatial resolution, think about enriching in the enriched fractions, but obviously these things come from either different sample types, tissue types, cells and cultures, et cetera. And the diversity of those different sample types actually often play a role in how you think about writing the applications. The nice thing about it is that allows us to apply these to lots of different biological questions. And don't get me wrong, understanding the biology of these questions is really hard. And I think it's kind of a key thing in writing effective grants. I will also say is I don't think I'm a particularly good grant writer. I was just looking through my NIH Commons just before this. I think in the last 18 years, I've written, I think I saw about 110 grant applications to the NIH, and I have about a 70% rejection rate overall. So that may not be bad if you're like a baseball player as kind of a hitting odds, but I think overall, part of the reasons why there were so many grants that have been written is because you just have to take a lot of shots on goal. And one of the great pieces of advice that I got from John Yates when I first started my lab was, one is to make sure that we have certain pieces of certain biological applications that we really understand the biology on, that we work on ourselves in our own lab. And then the second aspect is to make sure you're always juggling multiple different grant applications at the same time, and that you're thinking about how you're taking the results and the comments back from one grant application and potentially cycling it to another grant application later down the road. There's a lot of kind of nuances in grant writing. I think that people like Rebekah and Jesper might mention more specifically, and definitely happy to think about and discuss comments about our experience with it. But overall, I tend to think about these things as very analytically minded, and how do we apply these things to different sample types. So I think about it much more from the analytical side of things, which I must admit I think is hard in a kind of a biomedical research application. But anyway, that's I'm happy to answer other questions too.
Thank you very much for your insightful presentation. We'll move on to the next speaker, and we can leave the questions till the end. So our second speaker is Dr. Rebekah Gundry. Dr. Rebekah Gundry earned her PhD from Johns Hopkins University School of Medicine, where she focused on mass spectrometry of biomolecules. Following her doctoral work, she furthered her research during a postdoctoral fellowship in the Division of Cardiology at Johns Hopkins, but also at ETH Zurich, as well as the NIH. And her studies focused on cell surface proteins in stem cells and their karyogenic derivatives. Currently, Dr. Gundry is a faculty member at the Medical College of Wisconsin. Let's welcome Dr. Gundry. Thank you very much for being here, and we're eager to listen to your talk.
Thanks. So I am actually at the University of Nebraska Medical Center now. I moved here in 2019, and I'm a professor and chair of physiology, and I direct a Center for Heart and Vascular Research and the Mass Spectrometry Core. So in terms of grants, you're always going to get a ton of advice from people as you go through your career. I think it's really important to know a little bit about the people giving you advice, so you know kind of where we're coming from. For me, I only have experience writing grants in the United States. I've been a PI for 14 years. I didn't count the number of grants like Mike did. My institute counts dollars that I bring in. So it's, for me, my own lab, more than 30 million, but I've had a bunch of failures. And I think one of the things that I've learned that might be most relevant for this group, very much like Mike said, my lab really only focuses on questions in the heart, although my training really focused a lot on technology development. So what I've learned over time is that you have to understand how the game works, and you have to understand the rules. And one of the important things that you will find, at least in the United States, there's a whole lot more biology reviewers than there are technology reviewers. So what has worked for me is to never put in a purely technology development grant, because there's very few study sections that will like that. But I can come from the aspect of, to understand this question in the heart, I need to be able to answer this. And to do that, I have to have this tool available. And that's why I'm developing it or applying it. And so that's what worked well for us. All right. Again, you have to know how to play the game. I would say, make sure that you have thought about how study sections work and are structured. It can be a mystery depending on, but generally the process is people read the grant ahead of time and they come to study section and discuss it. How they behave in study section can certainly be a mystery, but the overall process is pretty much the same. I would suggest kind of looking at YouTube to see how it works. My rule that I teach people is that 3 must become 30. So generally 3 reviewers will read your grant. You have to have those 3 reviewers love it, but that's not enough. You have to make it so that those 3 reviewers advocate for you in front of a panel of 30 people to get all 30 people to be on your side. And that is very different. You're not just writing for one reviewer to like it. You have to make it so that a reviewer who may not be so familiar with your topic can be confident and be an advocate for you in a room, especially when there is somebody who doesn't like you in the room. So for example, you can have 3 reviewers, you can have, but remember that not all of the reviewers are going to discuss all of the things about your grant. Generally, the first reviewer will give the overview of your application, a summary, and what they thought about it. Reviewer 2 and 3 do not then give an entire summary of your application. They only pick out new things or if they have an opposing view. But everybody votes. It's not just the 3 people who reviewed your grant. So think about this. Think about personalities matter. And this is why you can put the same grant into 2 different study sections and get 2 different scores. You can have a reviewer 1 who loves your application, but isn't really an expert and is kind of a quiet, non-aggressive person. You can have a reviewer 2 that also likes it a lot, has some experience, but maybe is too not wanting to go to bat for you because there's another person on the study section that's more aggressive. And then you have a third reviewer who doesn't like it, is really loud, really aggressive. If you don't make it easy for reviewers 1 and 2 to go to bat for you and stand up to reviewer number 3 in that room, it's highly unlikely you're going to receive a fundable score. And there's nothing you can do about that other than make sure that your grant is written so that they can advocate for you. So remember the reviewers. You're going to get a lot of advice. Reviewers are the judge. Make it easy for your lowest common denominator reviewer to love it. So think of the worst case scenario reviewer, the one who doesn't really care. They spend minimal amount of time reading. They don't prepare ahead of time. They're constantly telling their program officer, oh, I'll get the reviews into you late, they're skimming it. And then a lot of times, if they don't love it by, page 2, the whole rest of the time they're reading your grant, they're just looking for ways to justify their low score. I know it sounds really mean, but it happens. Remember, the reviewers are scientists. We all have egos. Feed that ego. When you read something and it makes you feel like you understand it and you feel smart, for most scientists, that puts them in a good mood. Say, oh, I like this. I feel really smart. I want to read more. If your grant is not understandable to a scientist, they may feel stupid. And then they may not be in a good mood as they're reading the rest of your grant. So especially on your Ames page, but throughout, put your reviewer in a good mood, make them feel smart. General advice, write concisely. Everybody in my lab has to take this Coursera course. It's free. It's really helpful. More words is not better. Concise, accurate, clear language is really your friend when it comes to grant writing and grant review. I am going to give you all a copy of all these slides so you don't have to write down the details. Definitely use plain language. Don't use technical words. And if you're going to use a technical word, make sure that you define it. This is, again, going back to that lowest common denominator. Write what you mean and mean what you write. Make sure that you need every sentence and make sure that every sentence that you have is actually needed to tell your story. For me, I read things out loud to myself and I'll tell you why. When I'm done with the section or the grant, I print it out and I read it out loud. And this is why, because when you just read something with your eyeballs, your brain is really smart and it's smarter than you're expecting. It will fix all your typos. It will fill in the words that aren't there. It'll fix your spelling errors. But if you force yourself to actually read every word out loud, you can hear the errors. I know it sounds really painful. It can take time, but I promise you it will help you proofread. And the thing that drives me crazy the most is somebody that puts in a sloppy grant. Like, why should I spend my time caring about your research if you don't care for it enough to even write a well-crafted grant? Another important thing, connect the dots for reviewers. A lot of early career writers, I think, make a lot of assumptions about what they think the whole world knows. Nobody knows. So don't make any assumption about what a reviewer does or does not know. Well, everybody knows about heart disease. It's the #1 killer, so I don't have to introduce that. Actually, you do, because maybe a reviewer doesn't have expertise in that. Don't make the reviewer have to think and don't make them have to interpret it. Really spell it out. All your application pieces fit together seamlessly. Don't give them an easy reason not to fund you. You got the angry reviewer is just looking for, oh, you ignored these 3 papers from my lab, or these pieces are not well connected. You don't have the right references and your formatting is terrible. Remember the reviewers, most of us are not in our 20s anymore and haven't been for a while. Reviewer fatigue is common, so make it easy to read your grant with nice formatting, nice figures, nice line spacing, that sort of thing. Great, biggest advice I see for especially folks that come through kind of with omics and want to apply it to a biological application, I want to know why. And for most people, the why is much more important than the what or the how. Why are you picking this disease? Why are you asking this question? Why are you using this model? And make sure that you're telling them explicitly why, and don't make a reviewer guess as to why you made these decisions. When it comes to the Ames page, if not all grant applications, so again, this is at least in the United States, most of them require an Ames page. There's only one job of the Ames page, and it is not to describe your entire grant. It is not to give them all the details. Your Ames page, one job, and it is to get the reviewer to turn that page. Sounds really sassy, but there are reviewers who are not going to read your grant if they don't like your aims page. So it's your commercial. Again, you want your reviewer to feel really smart after reading your aims page, that they're like, oh, I get it. This is an important question. I'm so excited about it. Now I want to know how they're going to do it because they've gotten me interested with this advertisement on page 1. For most of your people who are voting, most of your 30 on your panel, this is the only page they ever might read. Don't give them all the details on the aims page. It's not necessary. Again, I'll send you all the slides. Don't include, certainly not details of kits and cell line names and things like that. Just big picture stuff. Again, why before how, especially for, you know, when you get into your aims. I see a lot of times folks tell me what the gradient is for their LCMS. Generally, this is not necessary or helpful in a grant application. Just cite a reference. I'm going to do mass spectrometry because that's what I do and I'm good at it. And this is why I'm going to do this approach. And here's what I'm going to learn. Again, a lot of rationale for every aim. I remind the reviewer why I need to do this aim in this way. Keep it non-technical. Expected results. Whenever I read a grant, this is always the thing that I will hammer the most. It is the most important section because this is where you convince a reviewer that you're going to learn something and succeed. Generally, we're not going to question, can you do a mass spectrometry experiment? We're going to expect that you can do that, but we want to know, what are you going to do with those data? And is it going to be worth the money? So for always do so much more than just tell me what you're going to do. Like, you can talk them through the method section. Great. We don't usually have a problem believing that you can do that unless it's something truly novel. You want to spend some time saying, yeah, we're going to make it work, but tell me what you're going to do with those data and how you will interpret it. And again, grab the reviewer by the hand, walk them through your thought process, because at the end of the expected results section, if this is written well, you will have convinced me as a reviewer that you've thought it through, you know what you're going to do, whether it works or whether it doesn't work, and that the money will be well spent in the end. And that is all I have for this. I will happily answer questions, and I will share these slides if you are interested in those.
Great. Thank you very much, Rebekah. That was really interesting, and I'm sure many people will have questions about that. But before we move to the questions, we have our final speaker, who is Professor Jesper Olsen. Professor Olsen studied analytical chemistry at the University of Southern Denmark, and then earned his PhD under the mentorship of Matthias Mann. He then did some postdoctoral research at the Max Planck Institute for Biochemistry in Munich, before returning to Denmark to lead his group at the Novo Nordisk Foundation Center for Protein Research at the University of Copenhagen. And he's also been promoted to full professor as of 2014. So yeah, we're really interested to hear what you have to say. Jesper, looking forward to your presentation.
Thank you, Rebekah. I'll just give a brief introduction to my group and some of the lessons that I've learned over the years. So I'm part of the Center for Protein Research here at the University of Copenhagen, where we have 5 research programs and 16 research groups, and altogether more than 300 people working here. And I'm now the vice director of the center, and I'm also running my own research group, where we are now currently roughly 30 staff members, including the people running the tech platforms. And in my group, we focus a lot on phosphoproteomics, deep proteome profiling, cell signaling, MS technology developments, single-cell proteomics, and also have a specialty in paleoproteomics. And with all these very wide range of interest areas, and with a lot of people in the group, we of course need to secure a lot of funding to keep the lab running. In Denmark, we have a little bit of a special funding landscape because we are fortunate enough to have the Novo Nordisk Foundation in the country. And this is actually the wealthiest charitable foundation in the world currently. And they are very good in providing grants for research and to researchers, particularly in Denmark. And annually, they are handing out more than 5 billion Danish kroner for scientific, humanitarian, and social purposes at the moment. And the center that I'm part of, we've been fortunate enough to get large center grants from the Novo Nordisk Foundation, actually several times. The center was inaugurated in 2007, and I've been part of writing grants for the center in 13, 18, and now again in 24. Unfortunately, they decided to stop the funding of our center with a phase-out grant towards the end of 2026. But I've been fortunate enough to be awarded a research laureate grant from the foundation to continue my group and the proteomics platform here beyond 2027. I've also been involved in a number of large Danish roadmap grants to get funding for equipment, and also a lot of other national funding agencies where I've gotten like individual PI grants from many of the different funding bodies that we have here in Denmark. But I guess more importantly, I've also been member of a number of large EU consortium grants, both as a PI and as a co-PI. So I have a lot of experience in writing these kind of like consortium grants. And I've also been fortunate enough to get an ERC grant as part of a large synergy application. And then I've also been actually a panel member of the ERC Consolidator grant panel for the last 4 years now. And that actually given me a lot of experience in both evaluating and scoring high-end applications to the ERC. So during my time here at CPR, we've been more than 200 million euros that we have been able to secure for running the center. And as a PI, I've gotten additional roughly 30 million euros to run my research group. I would say that there are 5 main things that are really interesting to consider when you write a grant. The first thing is to, of course, try to convince me as the reviewer of why the problem is worth trying to solve. So what is interesting and why should somebody care about this? Then the second point is to consider why are you the right person to basically solve this problem? Why are you, or maybe as a team, the best ones in the world to try to do this? It's also important to somehow convince us why is now the time is right to try to solve this problem. What has changed maybe in recent years that now makes it interesting to try to solve a specific question that could be, for example, new technologies that have become available. And what I would always recommend is to provide some preliminary data that will show some kind of feasibility in order to be able to demonstrate that you actually have the capabilities to solve the problem. And then lastly, it's always good to seek advice, either among your peers or maybe if you have mentors that can give you input on your grant applications. So that was my short pieces of advice, and I'll be happy to take questions as well.
Thank you very much, Professor Olsen, for your presentation. In particular, it's very interesting to see the national differences in the landscape of grant writing. So let's kick off the questions then for all the open for the panelists, and I'll open with one question that nicely ties the topic of collaborations and how to go about writing grants in a team. So, of course, collaborations often enhance grant proposals, but especially as an ECR, how do you find a good balance between collaboration and project ownership and balancing out the contribution of different collaborators? Maybe we can go around the table.
I mean, I can start. So I definitely think that this is a very different thing as in different stages of someone's career. So I will point out the piece of advice that I got when I was starting, I think was an excellent piece of advice, and that is when you go up for tenure and for your later things, you need to be able to point to what your major scientific contributions are. So when you actually have these grant proposals, it's not enough for you to have funding that's coming from lots of other people's ideas and major programs. You need to have things that are largely yours and driven by you. So when I actually started my lab, the main emphasis I wanted was I sought out the collaborators that I wanted in order to help me foster my goals and ambitions in my research. And you'll be shocked at how many people are really willing and excited to actually help you with that. And I also will point out those people became also really great mentors down the line, and actually helping me kind of define and providing feedback on early grant proposals as well, because they were often very familiar with how those grant proposals often would get reviewed and the types of study sections and reviewers that would seek them out. That being said, I do feel in most of my grant proposals, because I am an analytical chemist and I am a kind of technologist, do actually have some collaborators. There are some grant proposals which we do entirely within the lab. And that was actually something that John Yates also pointed out to me early on, was make sure that we have some projects that are entirely done within the lab that we can control, and that we're not actually necessarily being at the whim in some regards of our collaborators. And that's not being negative about the collaborators. It's just in some cases, those projects, your project may not necessarily be theirs all the time. So you need to have a certain number of projects where you can make progress on these even if the collaboration doesn't work out as quickly or as fast as you'd like. So those are at least my point. I think collaborations are really, really important, but you have to be able to point to what sort of things you're driving. As I've gotten more senior, I've found that I've tended to actually play that role that a lot of people played for me more often, where I am actually aiding other more junior people in their projects and helping them with securing their funding, where we're now playing kind of that more mentorship type role and support role. For some reason, I can't.
Yeah.
Oh, there you go.
Yeah, I'll echo that. Now in my role as department chair, it's the same. I'll give you the same advice I give new faculty, is that, as you, especially early, it's important that you get your own house in order and make sure that you're able to define what your research program is, for the same reasons that Mike said. When it comes to promotion and tenure, at least in academia, that's what we're talking about. That is absolutely critical. So it can be really challenging if you are writing grants with your postdoc advisor, where you're both principal investigator. It can make the P&T committee and your department chair have a really difficult time dissecting out what's yours versus, is your postdoc mentor just taking advantage of the fact that you're a rock star and they're gonna, ride your coattails, or are you getting more help and you're not as independent? So I think having collaborations inward versus outward is how I kind of think about it. As Mike said, what are the people that can help you? Those are the ones that I would focus on first. I would then, as you become more established, focus on the outwards. How can you help others? But don't focus so much on helping others at your early stage, because you really need to get your own house in order, get your own reputation out there, get your own, what do you own? Where are those senior author papers that are going to feed the next one, right? Like I said, think about setting yourself up for success, and that you don't want it to be in the situation where there's someone else who's really in control of your future, right? And if you leave all the biology to somebody else, you're always going to be relying on somebody else. You can set yourself up to have some challenges down the road when it comes to all things related to promotion and tenure.
Yeah, I can only second what Mike and Rebekah said. I think it's really, really important to basically maintain clear project ownership for the, although collaboration is obviously very, very important, but especially as an ERC, you really want to maintain the clear project ownership. For example, when you apply for ERC starting grants here in Europe, you basically have to list publications without your supervisor and postdoc advisor. So it's really, really important that you consider, to be able to demonstrate that you've been independent. And generally, when you collaborate with people, I think it's if you want to maintain ownership, then you should collaborate with people outside your own field who can basically add, value to your project. So it's absolutely not a good idea, at least not in the early stages, to, for example, collaborate with your postdoc advisor, because people would think that there will just be a continuation of what we've done in his or her lab.
Thank you very much, guys. That's really interesting. We had a panel about collaboration previously, and I think it was Vera, I can't remember exactly, or maybe it was John Yates actually, who said that the person with the least interest in the collaboration has the most control over how it turns out. So I guess that is true in the sake of grants as well. Just a reminder to everyone in the audience that you can put your questions in either the chat or in the Q&A, and that Jessica and I will read those out. So the next question follows on a little bit from what has been mentioned about preliminary data and expected results. So at what point in the stage of collecting preliminary data should you start writing the grant, or what order does that happen in, especially if it's something really, really new and you don't actually know the expected results? How much preliminary data do you think is actually necessary to get going with the grant writing?
Do we go in the same order, or okay, I can go.
Yeah.
So this is, again, very different from an early career person versus a more senior person. My recommendation to early career people is just start writing grants as early and often as you can. You will get rejections. Those rejections provide you with a lot of feedback, and you learn a lot from the experience and just in general. Just having all the, so most of my experiences through the National Institutes of Health, like Rebekah's, and there's a lot of just paperwork and standard documentations and forms. When you start to write lots of grants, those are very easy to put together because you put them together so many hundreds of times previously. So I really think that's my recommendation. But also, you have to realize that a grant proposal that have the exact same idea, and that it'll work just as well as another person, someone who has good preliminary data proving it will work, is going to be way more successful than those that don't. But there's also a delicate balance. I've also had grant reviews come back that said they've basically already done the project. So there is basically this amount of preliminary data that demonstrates that things are feasible and that there won't be any major hurdles to overcome, while at the same time still demonstrating that there's a lot of innovation and still to be gained. But I encourage people to just start putting together proposals early. There's a lot of work. If you haven't done it in the past, it takes a lot of time to get them first together. It gets easier over time, and just the more times you can practice, the better off you are. That's my feeling. But I don't downplay the time it takes to reread the grant proposal, like Rebekah said, making it just really easy to learn, getting it to your collaborators early on for feedback and comments. I've learned so much from just working with other people writing grants and seeing how they write grant proposals. I know this is like another question that's coming down the pipe, but the best way to learn how to write a grant is to work on a grant with other people who have a lot of experience with writing grants. You won't, as also Rebekah pointed out earlier, you'll get lots of advice on how to write a grant proposal. All of them will be very, very different. And what you often do is take little pieces of those different components that people have suggested that have worked well for you. Now I'll point out, I still feel like I'm learning a lot, but I also, when you read a good grant proposal, you know it. And there's those grant proposals. There are some grant proposals that just blow you away, and the way that they're organized and spelled out, and it doesn't matter what the topic is. Also, like what Rebekah pointed out, it could be on something you know nothing about. It's completely something that you didn't think you cared about, but in the end, you want to be an advocate and champion for that grant proposal because of the way it was written. The best comment you can get back is when the reviewers say, "This is an extremely well written proposal." That's often and so that's something you should shoot for.
So I have two rules for preliminary data. I either include the data because I need to demonstrate to a reviewer that the method is feasible in my lab. I found the first few times I submitted R01s, this was the thing that they wanted to see that I could do this in my lab. So I learned that I mean, I'd been making stem cells for years. They wanted to see that I could do it in my lab and not from my postdoc lab. So it's either feasibility or that I can do the technique in my lab, or it's preliminary data that isn't about a technique, but it supports the hypothesis or the aims. And so those are the only two things that I focus on for preliminary data. And so how I structure it is, if it's a method that I've published before and my name is last or kind of last, I usually don't put the data in the grant itself. I just say, we're going to do it as we've done before because we're really good at it. Something like that. And then if it's something new that, we developed a new approach or, had a new SureQuant method or something that hasn't been published, I may put a snippet of that in there. And then for supporting the hypothesis, that is basically you have to give them enough that justifies why you want to do what you want to do. But as Mike said, you don't want to go too much and show that you've done everything already. So it's a delicate balance there. And that's just kind of how I think about it. And just make sure that when you're putting your preliminary data in, you connect it to why you're putting it in there. That's probably one of the things I see the most. Folks chuck in data and then I have no idea why they're showing it to me. So make it clear. And I usually put my figure, data demonstrating that this approach is well established in the Gundry lab. Then the reviewer gets it. Oh yeah, then there's no question. Because again, you don't want to give them that low hanging fruit to say, oh, well, she doesn't know how to do this, so I'm going to give her negative points for that.
Yeah, I also usually, like to see applications with preliminary data, because that really demonstrates the capabilities of the people proposing the grants. But that said, I think it's also really important to understand the funding landscape and especially the funding agencies' guidelines, because some calls may actually not like to see a lot of preliminary data, whereas other grant calls, they may like to see a lot of data. So it's more like if it's a high risk, high gain type of project, or if it's like for a starting grant, or if it's more for a consolidator, then more or less preliminary data could be important to provide.
Thank you very much. In the same vein, I think I'll add one of the questions from the participants. So thank you very much to all participants throwing out some questions. We'll definitely include them. And this question is about including figures and explanations that simplify the workflow or the methods of the study design. So how do you estimate how important it is to show the spotlight on the methods, and if so, in which part of the grant is it most recommended?
Yeah, just like Rebekah said, write the grant to the lowest common denominator, assume that they won't read it, but they will look at your figures for sure. They will. And I know that that's a horrible thing to think about, that they're not going to read your grant proposal. But I think if you'll be more successful once you kind of accept that they may not read it. And what you want to do is to be able to get them very excited that they will read all the details. And the best way to do that is to have really pretty figures that they can get the main gist of it from the figures and from the, and again, in the NIH system here in the United States, it's a specific aims page that basically almost everybody on the panel will probably skim those at least when it's being discussed. So that's also very important. But yeah, figures are key. And also even with papers too, it's a really great thing in order to spend a lot of time on and get good at making figures.
And for the workflow figures, workflow of the methods, I think, if you're doing kind of standard bottom-up proteomics, digesting a protein and peptides, and, doing quantification, you may have a better use for that space than to draw a figure of that But if you're doing something that's really novel, like it's really important to get a point across to the reviewer of how you're distinguishing what you're developing or doing, which is different than what everybody else did before. And there's some parts about the method that make it unique, then definitely, take some time to put that into a figure, because at least in the United States, what most of our grants now have to have even a section that says the rigor or lack of rigor of the prior research. So you want to say, I know that research in this area has been done for 20 years, but actually all these methods have this huge limitation. So here's why the approach that I'm using is going to be different. And that can be a really good way to get across your novelty and your significance, as well as explain to a reviewer who may not be, familiar with what you're doing. And, be succinct about it.
Yeah, I absolutely agree that the figures are key to your application. Yeah, I guess we all know that a figure can say more than 1,000 words, so it's also really a good way for you to get away with not having to write a lot of text explaining, for example, novel workflows or things like that. So yeah, I highly recommend to spend a lot of time making the figures really nice and shiny, because that is definitely what can impress reviewers afterwards.
Thank you very much. Another question we've had is, what are your tips if you're writing a grant proposal which does have to rely on collaborators? For example, if you're relying for access to samples, obviously we have a lot of people doing clinical proteomics these days. How does that work into the grant writing process?
Yeah, so this is a common thing that's used done quite regularly. So one, you're going to you probably have them as a co-investigator. You'll also need to discuss with them what it will actually cost in order for them to provide you those samples. I know that it sounds strange to have samples that may have already been collected, but there is actually a fair amount of effort still it needs to go to kind of accumulating the metadata in a form that you want. Also, there are definitely some cases we've had where they have not necessarily been a co-investigator, but they're from like a repository, and you need a very strong letter often in that case to be able to provide it. But also you need to have them show some excitement about the project and the use of the samples with the way that you're applying it towards. So either in a letter, or that needs to come across somewhat in the grant application as well. There will in the United States, there is obviously human subjects, and I'm sure everywhere human subjects concerns about some of these samples, especially for new samples that are being collected. A lot of cases, what most people don't realize is a lot of samples that have been previously collected and they're completely de-identified. If they're not at your institution, you can often have them, the IRB, declare them non-human subjects research, which is actually kind of something that gets confused actually in study sections. That study sections will say there is human samples, but there's no human subjects portion. So what I've started to do in my grant proposals that have human samples but are not human subjects is I write a human subjects portion describing why it's non-human subjects research. Anything that you can anticipate somebody might get wrong in the study section, or they might be confused by the samples, you should actually address in advance. But that is something that most people are not aware of, that sometimes you can have human samples that are not human subjects research. And so I'm not sure if that answered the question exactly, but I'm sure, I know Rebekah has a lot more experience with this than I do.
Yeah, but you covered the really important things. I think
Rebekah, your microphone's very quiet on our end for some reason. Yeah.
Okay.
Yeah, there it goes. It's back again.
Sorry, I got a new laptop yesterday because anyway, and nothing is perfect. The letter that you read, I would always, if you have a collaborator that is the one supplying the samples, as in if you're not generating the samples in your own lab, my rule is I always get a letter. And that letter should always be something more than, I will provide you with samples. It's usually clinicians speaking about how this project will help in the clinic or something like that. And I do exactly what Mike does. I got so tired of people saying, oh, she's got human subjects, and no, I don't. So I just do the work for them. This is, again, hold your reviewer by the hand, take them through the thought process. I'm using samples that came from humans, but they're de-identified, they're non-human subjects research, but I actually fill out the form of human subjects to say that they're non-human subjects. It just saves everybody time.
Yeah, I mean, my advice to for collaborations as part of an application is absolutely to reach out very early to potential collaborators and make sure that if they are the ones who provide you, for example, with with the samples you are to analyze, that you get this discussions going very early on. Make sure that they, for example, have all the ethical permissions in place. If it's clinical samples, that you're actually allowed to transfer them to your lab to do the molecular analysis by, for example, analyzing proteins and potentially being able to discover new mutations, etc. That this often can be quite an obstacle if you realize too late that all these permissions are not in place. Yeah. So I would say generally reach out early, figure out if they need to be co-applicants or not, and if they are to be just collaborators, then of course make them provide like letters of support for your application.
Thank you very much for all the answers. So our next question is how do you go about how and when do you go about with starting writing a grant? So what are the key elements that you start working on and securing?
Are we always writing?
Yeah, I think that's somewhat true. I think what Rebekah said is there's always just you should always be thinking about what your next grant proposal is. Sometimes you're thinking about multiple different grant proposals. Sometimes we have discussions with this in our lab meeting about where we should prioritize the grant writing efforts and the things and what data and what that we do need going forward. That is something we have a little bit more, you have a little bit more liberty with when you have a lab that has been funded for quite a while, and you're able to then use the existing data you have in order to go forward with new data. When you're an early career person, you're basically taking stuff that you and ideas that you originated probably during a postdoc or during graduate school that you're trying to integrate. I will point out one advantage of actually having some collaborators is it sometimes provides that gap in expertise that, like Rebekah pointed out a little bit about when, needing to have preliminary data to say this may be something that she had done 100 times, but the reviewers didn't know it. So sometimes it's nice to actually have letters of support indicating that from somebody who may have that expertise. For example, when I started my lab initially, I was kind of combining methods that I had used in graduate school versus that in a postdoc. And the first comments that came back, they had completely forgotten about all of our papers we had done in graduate school, which is not surprising. And even though I would highlight in my renewal or resubmission proposals about this expertise and illustrate all the papers, then I got concerned. I'm like, well, maybe I have to ask my PhD advisor to be as a co-investigator to provide that expertise. And he gave me this very wise advice at that time. He said to pick actually one of the competitors at that time from our lab to act as a co-investigator on the grant. And that person actually knew me very well. We had been in a lot of meetings, and I wouldn't really call him a competitor. It's like as much as like Rebekah and Jesper and I are competitors in the same sort of space area. I mean, we will frequently ask colleagues to ask for letters of support. That's saying to provide documentation evidence that we have that expertise. So I definitely want to encourage people at least to think about that. And those letters of support, don't underestimate the time it might take to get them. And I know people may or may not be familiar with this, but sometimes you want to make it easy for those letter writers. So if whether or not it's either drafting something for them, giving them a bullet list of things that you need to include in the letter, everything you need to make it easy to get a letter to say the things that you need to help you with your proposal, the better off you are. This is something that I think my students and postdocs didn't appreciate early on when they're writing their fellowships or the proposals, but it is a little bit of an awkward process of trying to write letters for somebody else for yourself, but it actually saves everybody a lot of time and actually gets you what you need, which is a letter to say a certain thing.
In terms of like really early career, if you're a new assistant professor, my advice is, should you be writing a paper or a grant? Well, if you don't have any papers out where you're senior author and they're from your lab, I'm going to tell you focus on your papers, because it's going to be really hard to find that unicorn study section that's going to want to give you an R01 without any manuscripts from your own lab. So it's always great. I find it really helpful to write grants and papers and presentations at the same time, because I'm thinking about how to tell my story. Like, that's just how I'll go for, you know, months maybe without something. And if I have 3 things to do at the same time, that can really simulate the writing process. But especially early on, getting the things done that you need to, because if you continue to submit grant applications that don't get funded, and the criticism is that you really haven't established yourself as independent. And I will tell you, reviewers are not always going to tell you that in those words. They will find other ways to criticize your grant, but it's always going to be in the back of their mind that, you know, they have one paper, they have no papers from their lab yet, and they've been an assistant professor for 3 years. What are they doing? Right? Don't set yourself up for that. So if you're able to come in and really hit the ground running and get those senior author papers out pretty quickly, but I wouldn't sacrifice, you know, submitting an R01 or a really big grant in the first year. I wouldn't do that if you don't also have those papers. I would get your papers done first. As you go along, you're expected to do them both at the same time. But for the very beginning, get those papers out.
Yeah, I totally agree that as a newly started researcher with your own lab, you definitely need to first build a strong publication record before you start to write really big, ambitious grants. But I still think that, I mean, as soon as you have maybe a first few papers that you can build on, and you start to have ideas, and when you can really articulate, well, why a certain problem is interesting, and you also can maybe build it on some new observations or maybe some new technological advancements in the field, that then is really a good time to write and start writing in a grant application.
Great, thank you so much. I think we have time for one last question. I think this is a really good one. Do you have any tips about how grant writing differs if you're writing a grant for a highly computational project or a bioinformatics approach? Is this more difficult, since maybe it sounds more technical, it can sound more confusing? How do you go about securing funding for this?
Yeah, so I mean, this is a tough question, right? So I think there's two lines of thought. One is either the real innovation behind, there's got to be some really clever innovation in the computational strategies. A colleague of mine, Bill Noble, is kind of a master at this. He actually is really, really good at explaining very complicated computational mathematical concepts without having a huge amount of math in his grant. I'm not sure how he does it, but he's able to explain these very complicated topics in a fairly minimal amount of words. And to be honest, I don't know how he does it. He does a really great job. So there's this level of innovation that can advance the field substantially. And then the other aspect is, from what our lab tries to do, is usability and impact within the community. So tools like Skyline and Panorama. The main thing we focus on is our user base, our documentation, our testing, and this is a different type of grant. It relies less, not saying it's not innovative, but the reviewers don't think of it as the innovation the same way, but they think of it as being important as kind of the approach and the impact as being high because of the user base. And so I often find a lot of colleagues either not wanting to make their projects free or fully open source, or they'll have a web app and they don't want to open up to everybody just yet. But one way to increase the impact is to have users and to be able to document and track those. So those were things that we did early on with the Skyline project and Panorama and Percolator, et cetera, and is be able to track the usability, use cases and impact. So I guess there's two different ways. There's the broader impact, and then there's the innovation, and they both get reviewed slightly different. And either way, you should the innovation can be hard to document why something is very innovative. The best thing to do with learning how to write grants is to see how other people do it, who are good at it. As I said, for the innovation side, I have colleagues that I've learned a lot from, but I'm definitely not as good as they are at describing those sorts of things.
I don't have nearly the experience that Mike does, and I've never written a grant purely for computational. So, and we don't develop the big, everybody in the world uses our tools like Skyline. The things that we develop are really just catered to how to do some of the kind of the niche things that we do better that we think are useful to other people. So our bioinformatic approach to getting those funded has always been to piggyback it to a biological application, technology development, mass spec. We want to answer this question in the heart. We have to do this at the bench. We have to do this in the bioinformatics. And so that's how we've done it, and then it gives you a little more leeway to there isn't the expectation that you're going to include all that technical stuff that is going to turn some reviewers off. And so that's just how we've structured our grants.
Yeah, I mean, I also do not have a lot of experience in writing grants that are based solely on the computational or bioinformatics. But I've reviewed a number of grants recently in the context of the ERC consolidator grants, and we can really see that due to this big revolution now in AI and deep learning, that many bioinformaticians now want to apply for large grants for basically mining data sets that are already out there and available. And I think in order to really be convinced about grants like that, there really needs to be a solid rationale behind it, and it needs to be beyond just like what the standard intuition would be of what you can get out of deep mining data sets. So that's something to think about when you need to convince non-expert reviewers like myself for grants like that.
So I would like to add one last thing on this topic, if you will mind quickly, and that is, for those even doing experimental type work, you do have to think very carefully. I got reminded this by what Rebekah said, is in the analysis of the data. So one thing that will be criticized if you're collecting proteomics data is whether or not you have a good plan for analyzing the data. You might expect that you will get comments from a statistician thinking about it. Are the numbers adequately justified? Are you going to have enough power? Have you thought about the statistical power? A lot of experimentalists, including myself, often think, look, we can just do this and we'll make all this work out, and we just expect that to happen. If you don't have a very well thought out plan for how you're going to analyze the data, and that may require writing new tools, developing new ways of visualizing the data, sharing the data with other people, then that will be criticized. So most data these days in proteomics will have some sort of at least analysis or computational analysis question. And that's also a great place where you can have a collaborator who's a statistician or a machine learning person or something, et cetera, that can help you with the analysis of the data. Anyway, just want to kind of highlight that.
For sure. My rule is I always have a statistician on every grant, because that's not the thing that I'm the best at, and I know I need help, and then it makes it so there's no question that we're going to have an expert on our team.
Yeah, I mean,
Awesome.
some journals like Science Signaling, they even require you to provide the name and the email address of the biostatistician who helped you analyze your data.
Awesome. Thank you very much. I think with this we can conclude this incredible online panel discussion. Thank you to the speakers for being here and for your time in preparing and answering all these questions. Thank you for the participants who also contribute with questions and for listening. This is what makes panels worthwhile having. And just as a note, these online panels are always available on the HUPO website. You can find the link that's available in the HUPost, and hopefully we can see many of you at the World HUPO in Dresden. If you're attending, don't miss the chance of attending the ECR events. And thank you so much.
Thank you very much.